I'd like to thank the Society for this opportunity to present cross-sectional on research that I did. Today I'll be talking about ATDC and its role in specifically pancreatic cancer stem cell tumorigenesis. The classic paradigm of tumorigenesis is shown on the left, where cancer cells are heterogeneous, but all cancer cells are thought to be form tumors. Emerging evidence has suggested that malignant tumors are quite heterogeneous, and that they are composed of a small subset of distinct cancer cells that are responsible for tumor initiation and propagation. And these are termed cancer stem cells. These cells are termed cancer stem cells because, like normal stem cells, they possess the ability to self-renew and produce differentiated progeny. Now, to determine if pancreatic cancer stem cells really exist, our laboratory established a primary tumor xenograft program from either primary tumors or tumors expanded as low-passage xenografts in non-skin mice. And we sorted these cells based on a variety of cell surface markers. Cells were then implanted in mice, and in vivo tumorigenicity was assessed. We identified the pancreatic cancer stem cell population containing and expressing the surface markers CD44, CD24, and ESA, which typically represented 0.2 to 2% of all cancer cells within this tumor. We also found that these cells had increased expression of developmental signaling pathways and also possessed the properties of self-renewal and the ability to produce differentiated progeny. So far, we have analyzed over 20 different patient tumors, and although most pancreatic tumors that we have analyzed have a percentage of pancreatic cancer stem cells that range from 0.5 to 2%, we have seen a few tumors like the one depicted here that have a higher content of cancer stem cells. These tumors with an increased percentage of cancer stem cells demonstrated enhanced in vivo tumorigenicity, suggesting that increased cancer stem cell content directly correlates with biologic aggressiveness. The clinical relevance of identifying and characterizing these cancer stem cells is depicted in this diagram. It has been hypothesized that cancer stem cells are resistant to standard chemotherapy and radiation therapy. And if so, these therapies are likely to only target the non-tumorigenic population, but the cancer stem cell population will persist and regenerate the tumor. It is thought that treatments that target the cancer stem cells specifically, either alone in combination with standard therapies, will be more effective in tumor eradication. While this hypothesis has been postulated, little data currently exists to evaluate this. So we sought to determine if indeed pancreatic cancer stem cells are resistant to chemotherapy and ionizing radiation. And if so, what are the potential mechanisms by which this may occur? To address the question, xenografts of primary human pancreatic cancers were established in immune-compromised non-skin mice, as depicted here. And the mice were then treated with either therapeutic doses of radiation and gemcitabine. These dosing regimens that we gave are similar to those that we use in our clinic. And the mice were then sacrificed at different time points, and the tumors were analyzed for growth, cancer stem cell content via flow cytometry, and cancer stem cell function. The slide here above demonstrates some of the results of the radiation experiment. We gave 2.5 gray doses daily to mice bearing tumors times 4, with 4 different pancreatic cancer tumors analyzed, and with 7 animals in each of the control and radiation groups for each of the tumors that are depicted. Radiation treatment suppressed tumor growth as shown in the graph in the top left corner, with harvested control and treated tumors shown in the bottom left part of the slide. We observed an enrichment of the triple positive CD44, 44, and ESA positive population, with a peak of this enrichment occurring 48 to 1 week following the completion of the therapy. With time, the enriched cancer stem cell content slowly returned to basal levels, presumably because the enhanced population of cancer stem cells continues on and differentiates and regenerates the tumor once the treatment stops. A sample flow histodiagram showing the cancer stem cell enrichment in the treated tumor, which is up to 6-fold control in comparison to controls, is depicted in the bottom right of the slide. As with radiation treatment, we observed a similar clinical effect of tumor growth inhibition with gemcitabine therapy. And when gemcitabine was given at regular intervals for a 4-week period, the percentage of stem cells within the treated tumors increased compared to controls, with values increased up to 4-fold from the basal levels. A representative FACS diagram also is shown, demonstrating this fourfold enrichment in the bottom of the slide. To further demonstrate that cancer stem cells are resistant to ionizing DNA damage, isolated triple positive populations of pancreatic cancer cells and marker negative pancreatic cancer cells were subjected to radiation in vitro this time, and assays were performed for apoptosis. Now, the basal rates of apoptosis between the two populations did not differ. However, cancer stem cells were much more resistant to apoptosis 40 hours following irradiation compared to their differentiated counterparts. Of note, when we irradiated marker negative cells, this does not induce marker positivity. To determine if similar results were observed in patients, we analyzed several samples of pancreatic cancers from patients who underwent neoadjuvant treatment with chemotherapy and radiation prior to their surgical resection. We observed a significant enrichment of cancer stem cells within the resected tumors. In this patient's tumor, cancer stem cells can constitute approximately 23% of residual viable cells. To understand the mechanisms by which this DNA resistance or DNA damage resistance occurs in pancreatic cancer stem cells, we performed an after-matrix gene profiling analysis of pancreatic cancer stem cells and their non-cancer stem cells from pooled samples of 4 different tumors. Displayed here is a small sampling of the data with genes upregulated in cancer stem cells shown in the above the x-axis, and the gene downregulated shown in the below the x-axis there. As expected, there was a marked increase in the expression of these cell surface markers, CD44 and 24, in genes of cancer stem cells compared to their non-cancer stem cell counterparts. Interestingly, we found an upregulation of several genes important in DNA damage signaling pathways, including ATM, ATR, which functions as a master regulator of the DNA damage response. Another gene of interest was ATDC, which was upregulated, and we discovered that ATDC was one of the genes expressed the highest, up to 6-fold higher than pancreatic cancer stem cells. Our lab has been investigating the role of this gene, which should play, and we've slowly discovered that it plays a significant role in the tumorigenesis of pancreatic cancer.